This is ISCI 2001, Chapter 26, Mendelian and Molecular Genetics. The themes for this lecture include genetics and meiosis in the cell, single gene inheritance patterns, dominant and recessive alleles, Mendel's laws of heredity, and part B of this lecture will actually include the structure of DNA, so we will not actually cover the structure of DNA in this week's lecture. It will actually be covered on next week's lecture. Genetics, meiosis, and cells. Genes are the portion of DNA that determines a characteristic. Genetics is the actual study of these genes and how genes produce characteristics and how the characteristics are inherited. And Mendelian genetics are the laws describing inheritance patterns based on the research of Gregor Mendel. Anytime there are these definitions in the lectures, make sure that you know these definitions. Super important that you know them because you will be tested on them. So make sure that you know these terms. What you see here are artist rendition of genes. You can see the chromosome picture here. Chromosome is basically just a very super wound up, coiled up picture of, or coiled up picture of DNA. DNA, if it wasn't coiled up, it would be so long that it wouldn't be able to fit into our cells. So every chromosome is DNA that is super condensed and coiled into the double helix. So where you see the DNA here, this DNA, it contains your genes. So don't get bogged down and confused between what's the difference between a chromosome and DNA and genes. Okay, the terms are really, you can really use each term for one another. You just need to understand that whenever you see this structure here, you call it a chromosome. When you see the double helix structure, you call it a DNA. But the DNA carries your genes. Okay? When the DNA is super condensed and it forms this structure, it's called a chromosome. So it just depends on what shape it is in, and that's what determines what you call it. The gene is housed in the DNA. The DNA, when it is super coiled up and looks like this, it is called the chromosome. This is Gregor Mendel. He is referred to as the father of genetics. He was a monk, lived in a monastery. He was the one who first did all of the manipulations on pea plants. He took seeds from a pea pod and crossed purple flowering plants with white flowering plants. He took the pollen from each and transferred the pollen from the stamens of the white flower to the carpel of the purple flower. And what he found was all of the flowers ended up from the first generation being purple. And he found that was pretty interesting. And so from that generation, he crossed them again, and he found that after crossing them, the second generation, the white flower showed back up, even though all of the parents had been purple. And so he did some more testing, and over years and years, he's the one who came up with the terms of dominant and recessive and codominant and incomplete dominant, and we'll get into those terms in a few minutes. But basically, this is the man that we have to thank for starting all of our genetic engineering. So, when we use the term offspring, offspring refers to just children or descendants or progeny. The term locus refers to the location of a gene on a chromosome. Okay? A diploid cell refers to a cell in a sexually reproducing organism that has all the chromosomes. So in a human, okay, we have 46 chromosomes. So a diploid cell would be a cell that has all 46 chromosomes. These would be your normal body chromosomes. A haploid cell would be a cell in your body that only contained half the number of the normal chromosomes, which would be 23. So that would be your sex cells. In females, that would be the eggs, and in male, that would be the sperm. Remember, we talked about in our lab that your diploid cells are made through mitosis. Your haploid cells are made through meiosis. Single gene inheritance patterns, terms that you need to know. First one is allele. The allele is the alternative form of each gene within a population. So it's basically like the different color eyes, blue, brown, hazel, green, and every variation in between. Brown hair, blonde hair, all of the different skin colors. Is very fair skin to very dark skin and everywhere in between. Those are everything that codes for that. Those are your different alleles. Then you have your genome. The genome is the entire genetic makeup of an organism. Okay, so everything that is in one organism's genome is what makes them how tall they are, the color of their hair, the color of their eyes, the color of their skin, personality traits, natural born instincts, that kind of thing. That's their genome. Genotype, those are the genes present in an organism. We're going to be talking about this a little bit more in just a second, but if they carry the trait for being tall, they can either have this trait or they can have this trait. Whereas their phenotype is a physical characteristic of an organism, and if they are tall, we just say they're tall instead of showing their genotype, which would either be this one or this one. Now, if I just confused you with this mumbo jumbo right here, wait just a few minutes and I'll show it to you in a Punnett square, okay? Okay, so allele, I'm going to show you the chromosomes up here in the top left. Locus for the flower color gene is shown here, okay? So the loci, the locus. You got purple flower, white flower. These two chromosomes, these are called homologous pairs because homo means the same. Okay, homo means the same, hetero would mean different. Excuse me. Genotype is the gene. Phenotype is what you see. Okay, so I was talking about a while ago, if you were tall, phenotype would be what you see, it would be the tall, whereas genotype would be the letters that code for it, or the actual allele that codes for it. So here's homozygous, here's heterozygous. Alright, so once again, homo means the same, hetero means different. Homozygous, when an organism has a combination of two identical or the same alleles for a particular characteristic, two big A's or two little a's, okay? This would be your genotype. Okay, the letters show the genotype. Heterozygous, when an organism has two different allelic forms of a particular gene. And here again, big A, little a, that's your genotype. Okay. Dominant and recessive alleles. Let's talk about dominant and recessive. A dominant allele, okay, it will always mask the effect of other alleles for that particular trait. It's denoted by a capital letter. So a while ago when I was saying if it was tall, it would either show like this or like this. That's because tall is dominant as opposed to short. So big T would equal tall. whereas little t would be short. Okay, now if you're saying, well, wait a second, why doesn't short mean S? If you're comparing two different traits, whichever trait is dominant, you use the first letter of that word. Okay, so if you're saying, if you have two, let's pretend we're on Mars, and there are purple cows and green cows on Mars. Just bear with me. Purple cows and green cows. And purple cows are dominant and green cows are recessive. Okay? If purple cows are dominant, then we're going to use the letter P to stand for purple. Okay? So, what would stand for green? Okay? It would be little p. Okay? So, if we have a purple cow, it could either be two big P's or it could be a big P and a little p. It has to have at least one big P for it to show the dominant trait. Alright? In order for it to be green, okay, so both of those are purple. In order for it to be green, it can only have two little p's. And I'm showing it's a little p by putting this little curve in front of it. Okay? My big p's did not have curves in front of them. Okay? So, two little p's equal green. Okay? Alright, so I'm going to erase this real quick. Erase all of this. We're going to do another example. Okay? Maybe a little bit simpler example. Alright, let's say that we have, we're going to use letters that are more easily distinguished. Let's say that we have flowers. Okay? Red is dominant. Okay? So we've got red flowers and red is dominant. And we're going to cross them with white flowers and white is recessive. Okay? So if I said red is dominant, red is going to be a capital R. White is recessive. So red is dominant, it's a capital R. White is recessive, it's going to be a lowercase r. Because we always choose the dominant first letter. Okay? So, that's why we say it's going to be denoted by a capital letter. The dominant allele is. So, if y'all are hanging with me that far, recessive allele, when the recessive allele is present with another allele, has its actions overshadowed by the other. It is masked by the effect of the other allele. It's denoted by a lowercase letter. Okay, so here's our lowercase letter. Okay, we can use Punnett squares to show probabilities, and we'll talk about that in just a few seconds. But as long as you're hanging with me so far with the whole dominant and recessive when you're comparing two things, we're going to use the dominant first letter as the big letter, and the recessive one will use the lowercase letter of the dominant. Okay? Here's a picture with allele for blue eyes, which is recessive, allele for brown eyes, which is dominant. Here's individual A. So here's our first person. Let's call him John. Okay? He has one allele for blue eyes and one allele for brown eyes. So what color eyes is John going to have? If he even has one allele for that dominant color, he is going to show that dominant color. It doesn't matter if he has one of the recessive, because that dominant is going to overpower it. So John is going to show the dominant color, which is brown. Okay, so John is going to have heterozygous brown eyes, which is still brown. Okay? The way that we would write that is his genotype, okay? And blue and brown obviously have the same letters, but big B would be brown, little b would be blue, okay? Homozygous would look like this. Okay, because they have the same letters, either two big B's or two little b's. Those are your two homozygous options, or you could have heterozygous. And remember, with heterozygous, the letters are different, which means capital and a lowercase. So the letters are different. So it says he's heterozygous, which means his genotype is big and little. They're different. One's big, one's little. So his genotype looks like that. His phenotype is what you actually see, which is brown. Okay? Individual B. There's your loci or your locus for that. It shows that it's brown. It tells you that he is homozygous, or this person is homozygous. Okay? We'll call her Mary. Okay? Mary is homozygous. So if she's homozygous with two alleles for brown, then we know that she is going to be big B is her genotype, and her phenotype is brown. Alright? And then finally, alright, let's say that we have Trey. Trey has two alleles for blue. The only way that you can show blue is if both alleles are blue. So he is homozygous recessive, which means he is little b for his genotype. Okay, and his phenotype is blue. Cystic fibrosis is a deadly genetic disorder. 1 in every 20 people carry a defective gene for cystic fibrosis. It's the most common genetic disorder in whites in the United States, affecting close to 30,000 people. The cystic fibrosis, what it affects is it affects the mucus production in your digestive tract and also in your lungs. So it's very, very deadly. It can be stopped or slowed down with a lung transplant, a double lung transplant. But it's very, very hard to completely cure. But what happens is it is a recessive trait. So that's a good thing that it's recessive. It means that both parents have to carry this trait in order for someone to get it. It can be compared to sickle cell, which is a recessive disorder that is carried among African Americans, or not just African Americans, but blacks. Also found anywhere in the not just the United States, but the world. Something interesting though about sickle cell is anybody that gets sickle cell or that has sickle cell, they can never get malaria, which can save their lives if they live in Africa, because malaria is very rampant. So it may be a way that their body has found to fight malaria. So that's pretty interesting. But anyway, both of these are recessive disorders where both parents have to pass the trait to their offspring. If both parents carry the recessive trait, their child has a 50-50 shot of getting this disorder. Let me rephrase that. It's more of a 50-50 shot of at least being a carrier for the disorder. You're at least going to have, let's say if you have 4 children, one of the children will probably not be a carrier. 2 of them will more than likely be a carrier, and the 4th one will more than likely have cystic fibrosis. But the chances of you having a child with cystic fibrosis, if both parents have a recessive trait for cystic fibrosis, is not good odds. You don't have very good odds of coming through without at least passing the trait on. So you're either going to pass the trait on and your kids will either be a carrier, or you're going to pass the trait and your kid is going to have cystic fibrosis. Okay, so if you look here on the left, this is what I was talking about. You have a father that's a carrier, a mother that's a carrier. If you have 4 children, the likelihood of one of them not being a carrier or not having is only 25%. The other 75%, you're looking at least 50-50 of them at least being a carrier. The other 25% of the child having cystic fibrosis. So you're at least going to be passing on the trait, 75% shot there. And you can see over here where it says cystic fibrosis is a hereditary disorder characterized by lung congestion and infection and malabsorption of nutrients by the pancreas. Mendel's law of heredity states that Gregor Mendel developed a method of predicting the outcome of inheritance. He performed experiments with pea plants to show the inheritance patterns for certain characteristics, and he determined which characteristic traits were dominant and recessive. We've already talked a little bit about the flower color. That was the first thing that we spoke of when I talked about Gregor Mendel being the father of genetics. Other things that he looked at was the flower position, whether it was axial or terminal locations on the plant. Another thing that he looked for was whether the seed color was green or yellow, whether the seed shape was round or wrinkled, whether the pod shape was inflated or constricted, if the pod color was yellow or green, or if the stem length was either tall or dwarf, or tall or short. You could say it either way. So there's lots of different traits that you can look at, and you don't just have to look at one thing at a time. If you look at just, purple versus white, that would be called like a monohybrid cross. But you could also look at, how many of them had purple and white flowers and yellow and green seeds. You could look at those things at the same time and call it a dihybrid cross, because di means 2. Or you could look and see how many had yellow and green seeds and round and wrinkled seeds. And you could call those dihybrid crosses. So you don't just have to look at one thing at a time. You could look at more than one thing at a time. But obviously, the more things you look at a time, the more data you're going to have to sift through and look at, and the more complex it's going to get. Mendel's law of dominance states that when an organism has 2 different alleles for a given trait, the allele that it is expressed overshadowing the expression of the other allele is said to be dominant. The gene whose expression is overshadowed is said to be recessive. So Mendel's law of dominance is pretty easy. It's just reiterating what we know about the dominant trait. Whichever one is dominant is going to be the one that's shown. The one that's recessive will be overshadowed by the one that's dominant. And this is just once again showing that law of dominance. You've got big G and big W's. Okay, it's crossed with something that is little g's and little w's. We can come up with whatever word that we want to for that. Okay, so whenever they're crossed, the big g's and the big w's are always going to win. So this one, whatever big G and big W is standing for, is obviously going to be shown. The big G is going to be shown. The big W is going to be shown. And then finally here, the recessive trait will be shown. Mendel's law of segregation states that when gametes are formed by a diploid organism, the alleles that control a trait separate from one another into different gametes, retaining their individuality. Parental alleles of a gene separate at meiosis into gametes. And this is what I was talking about during a little lab today. If you have an organism that has 46 chromosomes, during mitosis, it's going to split and form two cells that still has 46 chromosomes. Well, that's all well and good, but if you're wanting to create sex cells, you don't want those sex cells to have 46 chromosomes. So it's going to go through another phase of mitosis, but when it's forming the sex cells and it's forming gametes, it undergoes meiosis. So prophase, metaphase, anaphase, telophase 1, prophase, metaphase, anaphase, telophase 2. And once it goes through the second phase, it will have broken down actually, and it will make 4 sex cells from the original 1, and they will all have half the number. And it's just stating in this law of segregation that when these gametes are formed by this original diploid cell right here, if this was our original diploid cell, that when the alleles that control the specific trait separate from one another into the gametes, they're going to retain their individuality, and they're just going to separate randomly. It's just going to be a lottery. If you have 6 alleles that are going to code for eye color, that's why we have so many different shades for eye color. Those 6 alleles are going to randomly separate. They're not going to say, okay, all the blue ones go this way, all the black or all the brown ones go this way, all the hazel ones go that way. They're just going to randomly separate and go into the individual gametes. They're not going to choose where they go. It's just going to be completely random. And here's a picture showing the male forming the sperm, female forming the eggs, and how they will end up crossing each other to form possible offspring scenarios. So where you have dominant gene, recessive gene, recessive gene, recessive gene, these were the possible gene combinations. This child could have gotten a dominant gene and a recessive gene. Of course, it would show the dominant gene. Same thing with this one. This one could show the dominant gene and recessive gene, so it would show also the dominant gene. Both of these would have received 2 recessive genes, so they would both show the recessive trait for that. So, just depending on which trait they got, they would show the recessive allele. Mendel's law of independent assortment states that members of one pair of alleles separate from each other independently from the other members of the other pair of alleles. In other words, if 2 different genes are separate chromosomes, where the one allele goes, it is not influenced by where the other goes. So basically it's the same as the other. It's a basic lottery. When the gametes separate, they're separating independently. It doesn't really matter where the other alleles are going. They're just randomly splitting when they're going through the process of meiosis. And this is showing the hypothesis, the independent assortment. You've got big R stands for round. Okay, little r stands for wrinkled. On the other side, you've got a big Y that stands for yellow. you've got a little y that stands for green. Okay, this is one of those crosses that I was telling you about. It's called the dihybrid cross, where you're crossing two things at one time. They're crossing a round yellow pea with a green wrinkled pea. These are the two gametes that they're crossing, and these are the individual assortment possibilities that they're going to get. The actual results support the hypothesis of the independent assortment. The standards that were covered for biodiversity and heredity, we were able to demonstrate the role of DNA in heredity, and actually this one is going to be covered more next week when we talk about the DNA and the RNA when we finish chapter 26, and it will be a really short lecture. There's only like 10 or 11 slides on that one, so it'll be about half as long as today's lecture. We'll demonstrate the ability to explain mechanisms for transmission of traits between generations and distinguish between inherited traits and learned behaviors. Please don't forget to do your discussion post, do a minimum of 3, and do your quiz. It's due by Wednesday, no later than 11:59 p.m. And have a fantastic week. I'll see y'all on Tuesday.
Made with Doodlecast Pro.